This is Miss Litton, and this is my wonderful APBio students in a review. Say hi. Hello. This is Chapter 9, the cell cycle and reproduction. I just want to remind you to do your highly suggested reading and thinking. I haven't written your quiz yet with that question in it, but I will. So make sure you do that. Overall, if we look at this particular chapter, you have three big categories really. You're talking overall the cell cycle, so you want to understand the cell cycle, big picture wise. And you want to, part of understanding the cell cycle is understanding how the cell cycle is controlled. We talked about things like cyclins and CDKs in the control. Remember, we made an analogy with an airport that you have to have two things. You have to be able to clear through, you have to have a what? Ticket. And the ticket is like the cyclin. And then somebody checks your, that's like the CDK, it binds to the ticket. I don't know another way, I don't have anything for that, sorry. But your ticket is the cyclin, which builds up. And it binds with CDK, cyclin-dependent kinases. You know kinase is an enzyme, and an enzyme is going to facilitate a reaction of some sort, right? It's going to facilitate the cell cycle. You know that it has to clear through checkpoints when you go into an airport. And the checkpoints are, did we do mitosis correctly? Did we do DNA replication correctly? And that's when you see things like the P53 gene. Remember, we had 2 issues with cancer. There are oncogenes. These are ones that are going to like turn the cell cycle to move on, more like in the cyclin wheelhouse, somehow affecting the CDKs or the cyclins and saying go forward with the cell cycle. And then the tumor suppressors, these are on the other end of the cell cycle, the checkpoints, right? And those checkpoints are making sure, oh, this, we didn't do DNA replication right, we didn't do anaphase right, so we better shut down this whole cell cycle. So it's the same patterns when we talk about a cell cycle and the controls that are there. We can relate that to cancer because that's absence of those controls in some way. Cell cycle keeps going. Remember, RAS is like stepping on the gas. The cell cycle keeps going. And we don't have a way, our brakes aren't working. Our like P53 gene to make caspases to stop that cell, to make it do apoptosis. Okay, so those are, the cell cycle control and cancer are connected in that way. And then this part right here is the details of the cell cycle. We know what happens during G1. What happens during G1 in the cell cycle? What? We're making more organelles because we went from 1 cell to what? 2 cells. We know what happens during the S stage. S stands for synthesis. What are we synthesizing? DNA replication. This is when we make our sister. We just can't see them yet, but they're there. The sisters are there. They're just waiting, okay? And then in G2, what are we making more of? Proteins. Then we move into the M stage, and the M stage consists of 2 things. It consists of the stages of mitosis as well as cytokinesis, or the stages of meiosis and cytokinesis, right? Because we have 2 divisions. So we just need to review what do things look like during each of those times. And then cycle back around, that's funny, back into like how do cancer cells look, right? You know, how are they different? What is some of their behavior? So if you keep in mind your big ticket parts, then just fitting in a few details here and there won't be as hard, okay? And when you look at your highly suggested reading and thinking, the first one is talking about why do we have these checkpoints? You know, what are some things that they check for? And then the second part is talking about 2 different kinds of cloning. So you're taking cells that are already differentiated and you're making them make either more cells in therapeutic cloning or reproductive cloning. You're making another whole organism. So that's the part about the highly suggested reading it and thinking. So let's jump into the cell cycle and the things that happen at the different stages. So remember, we can start anywhere, but let's start with G1. So what happens during G1 of the cell cycle? More organelles. Tell me again, why would that be a good thing to make it G1? Because we just went from 1 cell into 2 cells, right? So in G1, we've undergone mitosis, right? And so we're working our way back around through the cell cycle, and so we're getting ready. We better make some more stuff to fill up our cell. What follows G1? S. And what happens during the S stage? DNA replicates. And we don't see them like this, but you could tell me everything about them, right? These sister chromatids have the exact same DNA. When they become visible, they will be held together by a what? Centromere, right? And are my sister chromatids identical? Yes, my sister chromatids, because it came from one DNA strand, going from one DNA strand, it opened up, and now we have two DNA strands of the exact same thing. Are they going to stay the exact same thing throughout mitosis? Yes, the only time we mess with them is when we're doing meiosis, and when we do what? Crossing over. But do we even have to worry about that for this quiz? No, because that's chapter 10, right? So we're not even worried about that. Just set it aside. Set it aside. Remember, tomorrow's quiz is chapter 9, even though you've learned chapter 10, okay? You good with that? Are you relieved? Okay, even you understand. Meiosis is super easy. Okay? Then what happens during G2?
Proteins.
Make more proteins. Good. And then the M stage, mitosis and cytokinesis. Basically, we need to make sure that since we have two copies, we need to make our two cells and get a red one in each one, a blue one in each one, a brown one, and a green one in each one. Right? Okay. So then, knowing that, let's talk a little bit about those checkpoints that we discussed. Okay. So your yes.
Are the proteins that are made in G2 just like special proteins for mitosis and cytokinesis?
Probably, I would say predominantly.
Okay.
Yeah, but you could be making you're always making proteins, right? And let me just regroup those. G1, S, and G2 are part of one whole bigger group called interphase. Okay, good. All right. So when you control the cell cycle, there could be an internal signal, something that's building up within the cell, maybe you're making through some sort of metabolic activity, or it could be a signal you receive from outside that triggers you. And it's either going to stimulate a metabolic, it's going to speed it up or slow it down of some sort. And then we looked at this one right here, and we saw cyclins. You have to have the right cyclins to go to mitosis, and you have to go through the right cyclins to do the S stage. So when you're making more cells or more DNA, you have to have the right cyclins there. But there's more than two cyclins, right? Remember all the cyclins we saw and how they engage? So there's not just two. I'm just saying here's when you predominantly would have these two cyclins. And those are going to bind with the enzymes, the CDKs. And then where you see the stop signs, these are places where you're going to have like security. Are you going through anaphase, correctly? Did your chromosomes line up? Are you going through it correctly? Did you finish mitosis correctly? Did you replicate your DNA correctly? These are checkpoints. And this right here is where the P53 gene, if I'm going to know one checkpoint, this would be it. That P53 gene comes into play there. And here was another diagram. We talked about that with the cyclins building up. And then here is showing you all the different cyclins. So there's not just those two. Okay, and we said that if you can't, you want to stop it, you want to fix it, but if you can't fix it, then you'll destroy it. And when we destroy it, it's called what? Apoptosis. Do you want these questions? Okay. Yes. Yes. Let's just wait. Well, let me just give them a direction here. Why don't you be your favorite holiday? Did I do that one? I just do it. What do we care? Be your favorite holiday. Go ahead now. Go ahead.
So the cyclins are between G1 going into the S stage, and then
cyclins are all over the place. I'm just telling you key points when you're going into mitosis and when you're going into the S stage of interphase.
Okay, and then so the kinesis, the enzyme, do the cyclins bind to that to create the metabolic reactions that can
they're going to trigger something. They're activating that enzyme in some process of that cell to move forward in the cell cycle.
Okay, so the enzyme helps move forward in
cell cycle
stages. Okay.
You got it. All right, go, go, go. 4 questions. Easy peasy, lemon squeezy. A lot of this is a lot easier for you now that you've had the exposure of Chapter 9 and Chapter 10. You're used to talking about chromatids and anaphase. Yes, baby.
Is there a difference between the P53 gene and the P53 protein?
Yeah, the gene, P53 gene, codes for the P53 protein.
I thought the P53 gene was cancer.
No. If something's wrong with the P53 gene, that means something's wrong with your ability to make breaks, which can lead to cancer. Okay, did you guys listen to me? Listen to me. Don't say to me the P53 gene is cancer. Okay? Remember, we have two big issues with cancer. You have oncogenes, right? And you have tumor suppressor genes. Oncogenes, remember, and I'll show you a graphic with that in just a minute, but oncogenes are saying something about, they were proto-oncogenes, which were normal. These are just normal cell cycle genes. But when they've gone bad, like if a general went bad, it becomes an oncogene. So a proto-oncogene is normal, an oncogene is bad. We think oncogene, oncologist, doctor. And that basically is some type of gene that codes for a protein product that facilitates the cell cycle even though you're not supposed to do the cell cycle then. So it's like stepping on the gas, and the one you're going to think about is what? Name a gene for me that's named it. RAS, the RAS gene. It's involved in a lot of cancers. Second thing that has to go wrong for you to get cancer is you have to have the inability to step on the brakes. Your normal braking gene, stop, take, did we do everything right, is the P53 gene, which codes for P53 proteins. It's involved in several different reactions saying, wait, stop, let's fix it, we can't fix it, let's kill it. Okay? So cancer is when you have something wrong with your P53 gene, so you can't make any breaks. But P53 genes are just like proto-oncogenes. They're normal every day. Your P53 gene is working right now because none of you have cancer. Your proto-oncogenes are working right now because none of you have cancer. But both would have to go bad in order for you to get cancer, right? You'd have to have your proto-oncogene become an oncogene, and you'd have to have your tumor suppressor genes fail. Okay, and remember we said there are like over 100 different proto-oncogenes, but there's about 6 tumor suppressor genes, one of which is P53. Okay? So P53 isn't cancer, but if it's broken, you can get cancer. Do you understand the difference between that?
Yes.
Okay. All right, so let's look at these. Yay! 1. Anybody just lucky, not good? Anybody need me to explain it? Okay. Yay! 2. So easy. Oh! Okay, so let's talk about this. There's division of the nucleus. That is mitosis. Cytokinesis is division of everything else in the cell. It's those girls right there. Okay. So that mitosis is nuclear division. Yes, if you don't pass the checkpoints, apoptosis. Apoptosis. All right. So, when you look inside of a nucleus, some genes are active, some genes aren't active at the time. If they aren't active, you pack them up and put them away, right? You don't need these genes right now. That's heterochromatin. It's tightly coiled, packed up, very condensed, put away. That's heterochromatin. Euchromatin is out and about, and those are active genes. That's one way to look at a nucleus. But a nucleus would look like this during what stage of the cell cycle? That's right, interphase. Yeah, okay. So during interphase, because the chromosomes are condensed, right? You with me? Yeah. What's this right here? Nucleolus. What's going to happen at the beginning of prophase? The chromosomes are going to become visible. So it won't look like this. You'll see those condensed sister chromatids. Put it all together now? So this is interphase. We don't know. It could be in G1, it could be in S, it could be in G2 right now. We don't know, right? Just looking at this, you can't tell.
Trick question.
Are you okay? Don't forget about the obvious stuff, right? That's an obvious thing. This is a cell in interphase, okay? Now, when we move into yes?
So on the
Stop so I can hear.
So on the notes, heterochromatin and euchromatin are not in the notes. Are they
Right.
like a definition we need to know?
You will eventually know that. Am I going to ask you that on the quiz tomorrow? Probably not.
Okay.
Yeah. But you can always add these things in, right?
Yeah.
Because it's nothing really about it in your chapter right now, but it makes sense to teach it to you right now because this is the way a cell looks like during interphase. Okay, at the conclusion of interphase, we're going to move into mitosis. What are the stages of mitosis? Prophase, metaphase, anaphase, and telophase. Now they will tell you when you read study guides, you don't need to know all the stages anymore. But if you don't you've still got to understand what happens, and the stage gives you a name for what's happening, so you remember everything. So I find it still of value for you to know those names because you might use it in an explanation that you do, especially when you talk about mitosis and where our variety comes from. But that's a different chapter. Okay. So when we talk about prophase, that's when the chromosomes become visible. And remember we watched a video how the DNA coils on those protein spools? Remember that? We talked about histone and non-histone proteins. Histone proteins are like the same. They're like the spool, no variety. You just wrap the DNA around it. But there are also non-histone proteins associated with the DNA, and these are more regulatory. Remember how I said some DNA is packed up and some DNA is out and about? Heterochromatin and euchromatin? Non-histone proteins are the ones doing that. Non-histone proteins are packing up your DNA and unpacking your DNA. Okay? So this DNA right here, it's wound up on histone proteins all wound together to form nucleosomes, and those are twisted on each other so we can see them. Each of these sisters is identical. Each of these sisters consists of an old strand and a new strand. For us, how many of these will we have? 46. Because you have 46 chromosomes in all your cells. Now, I heard the number 23, and what you were thinking about was 23 homologous pairs. But pairs indicates 2. So if you have 23 pairs, you really have a total of 46 chromosomes. Your job in my arms, my legs, my mitosis is to take all 46 chromosomes, line them up smack dab in the middle, and separate the sisters. That's all you got to do. So we got to make them visible in prophase. We got to line them up in metaphase. We got to separate the sisters in anaphase and reform our cells in telophase and cytokinesis. That's our job. However many chromosomes you start out with, your 2 daughter cells are going to have the exact same number of chromosomes. Yes?
So in the beginning of interphase, your cell has 23 chromosomes, but after the S stage, it's 46.
No. Okay? Your cell, listen carefully. Your cell has 46 chromosomes. But as a result of the S stage, each chromosome consists of sister chromatids. This is a chromosome, but if it's separated, it's still a chromosome.
Okay.
So sometimes your chromosome consists of a pair, sometimes of a singleton. That will help you.
Okay.
Okay? So from anaphase, telophase, cytokinesis, G1, singleton. Move into the beginning of the S stage, cell singleton. Leaving the S stage, double. In G2, it's double. But you can't see it, right? Because it's all unwound. When it first appears, it appears double. Why? Because we're getting ready to do my arms, my legs, mitosis. So we need to have 2 copies of information because I'm getting ready to divvy up my copies. Yes.
They still consider chromosomes?
Yes. Yes. A chromosome that consists of 2 chromatids.
Okay, but when they're not paired, like when they're just the 46?
It's still a chromosome. That consists in 2 states. This is called a light, whether it's on or off. Still a light, right? So there's 2 states of a chromosome. It can consist of 1 or 2. You don't call it, you wouldn't say a chromosome that consists of a single chromatid. You would just say it's a chromosome. We have 46 chromosomes. But you could say, oh, at this point, all of our chromosomes have doubled. They've replicated themselves, so they consist of 2 sister chromatids because they have doubled at this point.
Okay, that makes sense.
Got that?
23.
23 homologous pairs. But even then, when you're talking about 23 homologous pairs, you really have 1, 2, 3, 4 copies of the information at that time, right?
Mm-hmm.
That's why if you have 4 copies of the information, that makes sense that you end up making 4 cells. Right?
Yeah.
Okay? So if I only have 2 copies of the information, right, I'm trying to get all the information in daughter cells, I'm lining up, I'm going to separate those 2, I'm done. But if I have 4, right, I'm separating out the homologous pairs and separating out the sister chromatids. But consistently, right now, you have 46 chromosomes. You have 2 different ways telling you how to build your eyes, potentially. You could have the way your mom told you and the way your dad told you. You could have 2 different ways to form your hair, the way your mom told you and the way your dad. And in chapter 11, we'll learn about the genetics and how that plays out. What's a dominant trait, a recessive trait, all of that. We're going to talk about how that plays out. But you always have, in all of your cells, 2 copies because we're diploid organisms. Right? We're diploid, so we always are in this state of having 2 bits of information for everything. But when you make your daughter cells, you want all those bits to get replicated and sent off in mitosis. So if you have 12 chromosomes, you'll copy all 12 and separate them. If you have 50 chromosomes, if you have 1,000 chromosomes, you'll copy all 1,000, line them up and separate them in mitosis. Whatever number you start out with, you're going to make 2 cells with the same number in it. Okay? Question?
Okay, I like having a hard time on visual.
That's okay.
So we have 46 of those in each cell.
Okay, so this is how it would appear during prophase, right? Remember, let me go back here. Okay, let me go back to the simple. Okay, how many chromosomes are in that cell? 7 chromosomes. Do you see the 7?
Yeah.
I didn't draw it with homologous pairs. Okay, so really there'd probably be 8 right in there, but I didn't draw it that way. Okay, so there's 7 chromosomes total in that cell, for sure, right? Okay, so now if I do the S stage, there's still 7 chromosomes. They just consist of, you can't talk right in front of her when I'm trying to explain something. There's still 7 chromosomes. They just consist of sister chromatids.
Okay.
Yes?
And those sister chromosomes are held together by
A centromere.
Okay.
Yes, exactly. Okay, now, so this is how they appear. Here's before prophase, here's at the beginning of prophase, how they appear, right? Okay, and so now, and this is how they condense. We talked about how you go from DNA all the way into a chromosome. So now when we talk about mitosis, here is how, though you would not see them like this, but starting at G1 and the beginning of S, they would appear like this, right? But following S, they really are all doubled. You just don't see that they're doubled until you enter into prophase, because that's when the chromosomes become visible. Right? Any more questions on that? What is the 2n number of this cell? What is the 2n number? This is chapter 10, but actually in here as well. 2n number is 4. The diploid number is 4. There's 4 chromosomes, right? What's the haploid number? 2n equals 4. 2n is just the total, right? If the 2n number is 4, 1, 2, 3, 4, what's the n number? 2, because there are 2 homologous pairs. So if you're going to make a haploid cell, you only want one of the littles and one of the bigs. If my 2n number is 10, my n number is 5. That would be in my gametes. If my 2n number is 46, my n number is That's how many chromosomes would be in my gametes. Right? My 2n number is 50. That means every cell in my body has 50 chromosomes in it. But when I make a gamete, my n number is only 25, because I want one gamete that has 25 and another gamete that has 25 to get together through fertilization and reestablish the homologous pairs, which gives me my diploid number again of 50, 25, 25, 50, where I have 2 bits of information controlling all my traits. Yes?
So a homologous pair would be like if the big blue and the big red got together?
They don't have to get together. They are a homologous pair because they're the same chromosome.
Oh.
It's a dad version of the chromosome and a mom version of the chromosome.
And when they get together...
In synapsis of meiosis 1, prophase 1, then they can do crossing over, but that's chapter 10. That's chapter 10. Okay? But chapter 10 is messing up your understanding of chapter 9, which is why I'm addressing it. Okay? All right? But remember, those have 2 different purposes. Chapter 9 is saying my arm is my leg, my mitosis. It's for growth and repair. It's copy, clone time. No variety in this. I skinned my knee. I need to repair my knee. Mitosis. Right? That's mitosis. I'm growing taller. I'm 7 years old. This year I'm 10 years old. I'm growing taller. Mitosis. My, oh my, meiosis has a totally different strategy. A totally different purpose. That's getting down to half the number of chromosomes, so you can take 2 halves and in special hugs make a whole. Right? Fertilization. Right?
It's special.
So, that brings us to the stages of mitosis. Okay? Stages of mitosis. In prophase, the chromosomes are starting to condense, forming those sister chromatids that are visible to me. And when I see them now, I can see what they've been doing during S stage. What have they been doing during S stage earlier? They've been replicating. Oh, I see what you did. I don't know what she's saying, but I'm going to ignore it.
That got intense right there.
Okay, come back to me.
No, she's thinking about something else.
Here I am, here I am. So I can tell that the S stage happened because when the chromosomes appeared, they're all doubled. So I know they replicated themselves. And I'm going to break down my nuclear envelope so my microtubules can attach. So I got to get rid of my nuclear envelope. Nucleolus is going to break down. And so my chromosomes are visible. Oh, there are my centrioles moving towards the poles. The spindle fibers are forming. There's an aster right there where it's shooting all around out of the microtubule organizing center, MTOC, right? And then when I go into metaphase, everybody lines up single file. My arms, my legs, mitosis is single file. Single file. You only line up with a pair when you're getting ready for meiosis. We pair up for, right? So pair up. Pairing up is always having to do with sex. Right? Okay. But this isn't. This is more my arms, my legs, mitosis. This has nothing to do with sex, so we stand single file. Right? The whole time. Now we're going to separate. Yeah. I've got a copy of every chromosome now for both cells. And the nuclear division is focusing just on what's going on with the chromosomes and the reforming of the nuclear envelope. I got the nucleolus getting back that. Then this division of the cytoplasm is cytokinesis. The variation on cytokinesis is going to depend whether I'm a plant cell or an animal cell or a fungal cell, really. Why? Because some cells have cell walls. They can't do cleavage furrows. They build a plate, right? So let's go over the stages of mitosis. The whole thing's called what? Mitosis. First stage is prophase. The chromosomes, these are sister chromatids. Before S stage, it was just this. After S stage, it became this, right? Okay. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase. Away at... Who's going away? Good. Away at anaphase to nuclei at telophase. Cytokinesis. Okay? Yes. Except here where we're building a cell plate. Right? And growth and repair, right? So, here you go. Eight questions. Hurry up, because I need to talk to you about cancer. Yes, ma'am.
Do the centrioles, like create the asters, or is it from the nucleus disappearing?
The centrioles don't even need to be there because plants don't have them, but they just have microtubule organizing centers. Our centrioles just sit in our microtubule organizing center. It's a mystery.
Okay, so they don't have any part in...
Okay. No. All we know is they have that 9 groups of triplet, which can be found anchoring cilia and flagella at the basal bodies. It's odd. Let's go, let's go, let's go. Okay, I'm moving forward. Sorry, you're not done. Perfect. Question? Yay. Question? Okay. Oh, when are proteins being synthesized G2? Is that an application level question or a recall question? Recall. Those are some stuff you just have to know. Okay, that's a total recall question. Lucky, good. What were you? Good? I want you to be good, not lucky, but if you're lucky, I want to help you. Yes, exactly. Lucky or good?
Good.
Oh. Nuclear envelope breaks down. Chromosomes become visible, the nuclear envelope and the nucleolus disappear, radiating microtubules, right? Is that a recall or an application?
Recall.
Recall. Ah. Recall or application? Recall. You just need to know organelles are formed during G1. Logically, to remember that, think what came right before G1? Mitosis. So you were one cell, now you're two. Makes sense, you need to make more organelles because you're going to get bigger, right? Okay. Ah, this one, yeah. Recall or application? Recall. What is interphase? What does the S stand for?
Synthesis.
And what are you synthesizing? DNA. DNA synthesis. Oh, now I get it. Okay, 2 cells, nuclear envelope reforming, telophase. 2 nuclei at a telophase. Okay, that's it. All right. Now, here we go. Good cells versus bad. You lose a couple of things. You have abnormal growth, which means you have failure. Proto-oncogenes have become what? Oncogenes. And nobody's stopping them, which means failure of what? Tumor suppressor genes. That's how you get cancer. Now, normal cells have density-dependent inhibition, which means density dependent. If the density is too high, that inhibits them from making any more cells. They're not crowded here, so they're fine. Now we're crowded, they don't make any more cells. If you remove some cells, there's a space. They'll fill it in. Density dependent inhibition is a good thing. It's being able to sense that you are crowded. Another thing is they have anchorage dependence. Most cells need to be attached to something. If they don't have some substratum or something to be attached to, that's it for them. Are there exceptions to the rules? Yes. Red blood cells, white blood cells, right? Okay. So when you have a neoplasm or an abnormal growth, they do not care that they are crowded. They are ignoring that. They are not exhibiting contact inhibition. Okay, that's one of the problems with that. So they'll just keep piling on. So if it piles and piles on and doesn't spread out of a localized area, if it stays encapsulated, then you have an abnormal growth still, but it's just benign. You go in and get it removed. Why? Because it could go from being benign to being what? Malignant. What does it mean to be malignant? You have the ability to spread. You are not contained anymore. And when it does that, it metastasizes. And so something is wrong with your cell cycle. You're defying the normal regulations, and you've gained the ability to invade other places. We looked at what does a cancer cell look like versus a normal cell. Now, you know what codes for all your proteins? DNA. So does it make sense that you do, does it make sense then that you have abnormal nuclei? Yeah, because that's the stuff that's coding for your protein. So obviously your chromosomes are screwed up. They don't look like normal cells. They have little bleeds on them, little blisters on them. They don't look normal. Their DNA isn't normal. What does this mean? Do not undergo apoptosis. Yeah, they're immortals, right? They're immortal cells. No contact inhibition. They don't mind that they are what? Crowded and close. They are disorganized. They have multiple layers where normally you would not do that. They undergo, if they metastasize, what's angiogenesis again?
They create a vessel.
Create blood vessels to grow towards them.
What's the difference between density dependent and contact inhibition?
Okay, so same thing, just two different ways of talking about. Normally you exhibit contact inhibition. That's a property you would exhibit. And you're density dependent, meaning that I am density dependent because I exhibit contact inhibition. That's how I would say it. Okay, took me a minute, but I figured it out. All right, so we know that they look different. We know what angiogenesis is. Come here, little blood vessels. Why is that so bad about cancer that they can say, come here, little blood vessels, angiogenesis, beginning of blood vessels?
Easier for it to spread.
Yep, it can hit your ride, and it can also get resupplied. Hit your ride or get resupplied. Okay, I'll keep going here a little bit in case you need some help. Two categories of genes. Proto-oncogenes. These are genes that say go cell cycle. RAS, like stepping on the gas. Okay, so they pretend like their gas pedal is down even though it shouldn't be. They step on the gas even though it's not supposed to. Even though you haven't stepped on the gas, your car's still going forward. Something's broken. And then tumor suppressor genes, if something's broken, you want to step on your brake. That's what tumor suppressor genes code for, brakes. But if you have a problem with those, like a P53 gene, you're not braking. You're not stopping and going, hey, what's the problem here? Let's fix it. Let's kill it if we can fix it. Your brakes just don't work, and you get the crash of cancer. So we said that there are about 100 different proto-oncogenes that can become bad oncogenes. It's like 100 different generals that can go bad. And we talked about RAS as being an example of that. Because normally you have to be triggered by a growth factor to turn on RAS. But RAS doesn't even care if it's triggered or not. It just says, let's go. Let's make some more cells. This is awesome. Party on. Okay? So that's a problem. That's a bad RAS. And we talked about BRCA1, BRCA2 genes. And saying, yeah, let's just keep growing. Let's grow some tissues in here even though we're not supposed to. The other one, tumor suppressor genes, they're saying, stop. We need to not do this. And our big example is the P53 gene. If we don't have the brakes anymore, it's like going into the airport and not having any security. You just bring, nobody's checking to see if there's something wrong in your bag, you got a gun in your bag, you got a bomb in your bag, if this has gone wrong, they just let you go through on the airplane. Okay? That's what they're supposed to do. Stop, assess the situation, fix it. I'm going to remove these bottles or tubes or this gun or this knife out of your bag. Hi! Before I let you go on. I love you. I missed you so much.
You guys are lucky to have her as a
Yeah, you
teacher.
guys appreciate Miss Linton.
Yeah, thank you. They appreciate you too. All right. So here, look right here. So here's a P53 gene. P53 gene, remember caspase? What does caspase do? Hello, Mr. Jones.
Carry on.
Say hi to Mr. Jones. Everybody's coming by to say hi.
Hi, Mr. Jones.
Hello, everybody.
All right. Bye. Sorry. I'm writing physics curriculum, so both the physics teachers are coming by to help me, and of course I'm not available right this minute. Okay, origin of cancer. Either you're not fixing, or remember telomeres. Telomerase is an enzyme that keeps building your telomeres, and normally that tells you they get smaller, smaller, smaller every single cell division, and they keep rebuilding that chromosome. Okay, what else? The only other thing you needed to know then was in this part was prokaryotic cell division doesn't do mitosis. They don't have linear chromosomes. They're just a single circular chromosome, so it replicates, the cell separates. Binary fission. Okay, got it? Are you stressed? Yeah. No. You'll get this. I'm already proud of you. Okay, be smart.